Sir Godfrey Malata and Madam Rita brings you questions and answers. Biology.
Question number 1. You are provided with 2 maize seeds, labeled A and B. A laser braid or a scalpel and a ruler. Let's study the specimens and answer the questions. Question A. State 2 differences between A and B. Specimen A is a soaked maize seed, while B it is not soaked.
A, it is soft and large, while B, it is hard and smaller.
Question B. Using the razor blade or a scalpel, cut the longitudinal section along the narrow side of specimen, as shown. Draw the longitudinal section and label 3 parts.
Pericarp.
This is the outermost layer of the seed.
Endosperm.
This is the part that is found between the pericarp and the cotyledon of the seed.
Cotyledon.
This is the part that stores food for the seed.
Plumule.
This is the part that grows into a shoot.
Radicle.
This is the part that grows into a root. Calculate the magnification of your drawing and show your working.
Magnification is equal to size of the drawing over size of the specimen.
Question C. Explain why a germinating seed tastes sweeter than ungerminated seed.
It is because stored starch is now reconverted to glucose, which is sweeter starch by enzymes.
Question number 2. You are provided with a specimen labeled M, which is an onion, and a ruler. Question A. What type of storage organ is specimen M, which is an onion?
Leaves or bulb.
Question B. Make a biological drawing of the cut surface of the specimen M, which is an onion, and label any 4 parts.
Foliage,
which is found above the ground, and they are green in color.
Terminal bud.
Terminal bud, it is found in the inner part of the onion.
Leaf base.
These are the bases where the leaf starts from.
Roots.
These are the parts which grows into the ground. Calculate the magnification of your drawing.
By using magnification formula, magnification is equal to size of the drawing over size of the specimen. By using the ruler, measure the size of your drawing and the length of the specimen given, and then calculate the magnification using the formula.
Question C. The following investigation was carried out on specimen M, which is the onion. A sample of specimen M was crushed in a mortar. The resulting mash was pressed in 3 test tubes and water was added. The mixture was stirred, and the test tubes were labeled A, B, and C. The 3 test tubes were treated, and the results of the investigation were as follows. In test tube A, iodine solution was added. The result of color was brown. What is the conclusion?
Test tube A contains glucose.
In test tube B, hydrochloric acid and Benedict solution were added. The mixture was heated. The result of color was brick red. What is the conclusion?
Test tube B contains sucrose.
In test tube C, Burette solution was added. Result of color was blue. What is the conclusion?
Test tube C presents the absence of proteins.
Question number 3. You are provided with specimen J and K and a ruler. Specimen J is a soaked bean seed, while specimen K is an unsoaked bean seed. Question A. Write down the differences that you can see between the specimen.
Specimen J, it is smooth and big, while specimen K, it is small and wrinkled.
Question B. Find the differences in length between the seeds in specimen J and K. Describe what you do, your results, and your conclusion. 1. What was done?
The measurement was taken from the tip of the base of the seed, and the results were J was 8 mm, while K was 5 mm.
Question number 4. You are provided with a wing feather of a bird, labeled specimen A. Question A. Draw specimen A and label any 3 parts.
These are: number 1, quill.
This is the part which is attached to the skin of the wing.
Number 2, shaft.
This is the part that is found at the middle of the feather.
Number 3, vein.
This is the part which is attached to the shaft of the feather. Measure the length of your drawing and calculate its magnification.
By using magnification formula, magnification is equal to size of the drawing over size of the specimen. Using the measurement you have taken, find the magnification using the above formula.
What is the main use of specimen A, which is the feather, to a bird?
The upward and downward beating of the bird's wing to aid it in flight.
State 2 ways in which specimen A, which is the feather, is adapted to its function.
It has a quill, which is hollow and light, making it lighter. It also has hair-like structures called barbs, which further branches into barbules, which have hooks and ridges. These hook together to form an L4 that generates lift in bird's flight.
Question number 5. You are provided with a mango twig. Question A. What type of leaf arrangement is displayed by the twig?
It is a whole leaf arrangement.
What is the advantage of this arrangement of leaves on the twig?
This type of arrangement exposes each leaf to the sunlight.
Question B. Detach one leaf from the twig. Draw the leaf and label any 3 parts.
Number 1, petiole.
This is the part that is attached to the main stem.
Number 2, lamina.
This is the greenish part that is visible on the leaf.
Number 3, midrib.
This is the part that runs from the petiole to the apex of the leaf. Question C. Calculate the magnification of your drawing and show your working.
By using magnification formula, magnification is equal to size of the drawing over size of the specimen.
Question D. State 3 adaptations of the leaf for photosynthesis that can be seen in the specimen.
These are: number 1, it has thin lamina, which provides a short distance for diffusion of gases. Number 2, it has a broad surface that increases surface area for gaseous exchange. Number 3, it contains chloroplast, which is witnessed by the green color. This chloroplast traps light energy for photosynthesis.
Question number 6. You are provided with the following specimens. Number 1, maize seed, labeled specimen K. Number 2, sorghum or millet seed, labeled specimen L. Number 3, bean seed, labeled specimen M. Number 4, groundnut seed, labeled specimen N. Question A. Apart from color, use one physical characteristic to put the seeds into 2 groups. Write down the physical characteristic you have used and put the seeds into 2 groups.
The seeds have been grouped according to size.
Question B. By using the letters K, L, M, and N to represent the seeds, construct a simple dichotomous key to that can be used to identify the seeds.
Number 1A. Seed with very small size.
Millet.
B. Seed with big size.
C2.
Number 2A. A seed which is in a bean shape.
A bean seed.
Number 2B. Seed which is not in a bean shape.
C3.
Number 3A. Seed with a hard taster.
Maize seed.
3B. Seed with a soft taster.
Groundnut seed. Question C. State one way in which groundnuts contribute to the nitrogen cycle.
Groundnut plants have roots which contains nodules that fix nitrogen into the soil by their symbiotic relationship with bacteria.
Question number 7. You are provided with the following materials. Number 1, specimen X, which is Irish potato. Number 2, salt solution of the following concentration: 0%, 25%, and 50%. Number 3, a laser blade or a scalpel or a knife. Number 4, a ruler. Using a scalpel or a razor blade or a knife, peel the specimen and cut the specimen in small pieces that are in 2 cm long, 1 cm wide, and 0.5 cm thick. Place one piece in 0% salt solution and the other piece in 25% salt solution and the third piece in 50% salt solution. Leave to stand for 10 minutes. Remove the pieces and measure the length of each and every piece. Then try to bend each piece to test its flexibility. Finally, record your results.
The length of the potato in 0% solution was 2 cm, while its flexibility, it was stiff or difficult to break. The length of the potato in 25% salt solution was 0.9 cm, while its flexibility was a bit limp or easy to bend. The length of the potato in 50% salt solution was 0.32 cm, while its flexibility was limp or very easy to bend.
Question B. From the table, state the relationship between salt concentration and the length of the potato piece.
As salt solution is becoming more concentrated, the length of the piece of potato is becoming smaller.
Question C. Explain the results observed in the flexibility of the potato piece in 0% salt concentration and 50% salt concentration.
Potato pieces put in dilute salt solution became stiff and readily bent. The cells are said to be touched. Those put in very concentrated solution became limp and readily bent. They are said to be flaccid. What actually undergo touched and flaccid are tissue cells of the potato pieces. Through osmosis, water will enter the plant cells when surrounded by a dilute solution, while water is lost from a plant cell when surrounded by a very concentrated solution.
Question number 8. You are provided with a small fish. Question A. Draw the fish and label any two locomotor structures.
Number 1. Dorsal fin.
This is the part which helps the fish in swimming.
Number 2. Pectoral fin.
This is the part which helps the fish in changing directions. Question B. Calculate the magnification of your drawing.
By using magnification formula, magnification is equal to size of the drawing divided by size of the specimen.
Describe the shape of the fish.
Fish has a streamlined shape.
What is the advantage of this shape to the fish?
The shape prevents the fish from experiencing the force of drag.
The force of drag is a force which resists the motion of the body with fluid. In this case, it's water. How does drying preserve the fish?
Drying preserves the fish by preventing multiplication of microbes, as microbes cannot multiply in dry condition, but in a moist condition.
State 2 nutrients that human beings get from the fish.
These are proteins and calcium.
Question number 9. You are provided with specimen A and B. Specimen A is an onion, while specimen B is an Irish potato. Question A. Which one is a stem? And give the reason for your answer.
Specimen A.
Which is an onion?
It is a stem because it has roots down and leaves appear on the ground, so that the stem it is in the middle.
Question B. Apart from shape, state 2 structural differences between specimen A and specimen B.
Specimen A, which is an onion, has modified leaves used for storage, while specimen B, which is an Irish potato, has tubers for storage. A, which is an onion, has scales, while B, which is an Irish potato, has no scales.
Question C. State 1 similarity between the specimen A and B.
Both onion and Irish potato can be replanted using bulb and tubers.
Question D. How would you test specimen B, which is an Irish potato, for starch?
Starch can be tested by peeling the Irish potato using a razor blade or a scalpel. Then crush it using a mortar and a pestle. Add 2 to 3 drops of iodine solution using a dropper. Then observe the color change. The presence of starch it is indicated by blue-black color.
Question number 9. You are provided with a bean seed, which was soaked in water. Question A. Measure the longest axis of the bean seed and record its length in millimeters.
Measure the bean seed from the bottom to the top.
Question B. Suppose the bean seed was drawn to magnification of 4. Calculate the length of the diagram.
By using magnification formula, magnification is equal to length of the drawing divided by length of specimen. By using magnification formula, magnification is equal to length of the drawing divided by length of the specimen. Then make the length of the drawing as subject of the formula. Therefore, length of the drawing is equal to magnification multiplied by length of the specimen.
Question B. Using hands, peel the seed and separate the cotyledons so that the embryo is seen attached to one cotyledon. Draw the cotyledon with the embryo and label any 3 parts.
Plumley.
This is the part that grows into a shoot.
Radical.
This is the part that grows into a root.
Cotyledon.
This is the part that stores food for the seed. Question number 11. You are provided with the following: a very dry groundnut seed labeled specimen B, a very dry maize seed labeled specimen G, a ruler which can measure in millimeters, a matchbox with 10 sticks, a paper clip. Question A. Measure the longest axis of each specimen and record your results in millimeters.
By using a ruler, measure the longest axis of each specimen.
Question B. Draw specimen B with a magnification of 5.
By using magnification formula, magnification is equal to length of the drawing divided by length of specimen. Then make the length of the drawing as subject of formula. Then substitute the formula with the digits. By using the length calculated, draw the specimen on the paper.
Question C. Pierce the paper clip into specimen B, which is groundnut. Light a matchstick and use it to bend the specimen and observe what happens. The paper clip into the soft part of specimen C, which is a maize seed. Light a matchstick and use it to bend the specimen. Observe what happens. Record your observations for specimen B and C.
In burning specimen B, which is groundnut, oil oozes out. In burning specimen C, which is maize seed, soot is produced.
Question D. Explain your observations.
In burning groundnuts, oozing of oil indicates presence of lipids, while in burning of maize seed, soot was produced. This indicates the absence of lipids.
Question E. Describe how you can test specimen C for lipids, which is a maize seed.
Lipids can be tested by crushing the food stuff, which is a maize seed, using a mortar and a pestle. Smear it on a plain paper. Then observe what happened. The translucent spot show presence of lipids, while the absence of translucent spot show the absence of lipids.
Question number 12. You are provided with specimen X and Y. Specimen X is a sisal, while specimen Y it is a star grass. Question A. Draw specimen Y and label any 3 parts. Specimen Y it is a star grass.
Apex.
This is the pointed part of the leaf.
Lamina.
This is the part which looks greenish on the leaf.
Vein.
In this case, this is the part that is running from the sheath to the apex of the leaf. Question B. Calculate the magnification of your drawing.
By using magnification formula, magnification is equal to length of the drawing divided by length of specimen.
Question C. Which one of the 2 specimens could withstand dry conditions?
Specimen,
And explain your answer.
specimen X, which is sisal, because it keeps more water in its cells.
Question D. Give any one product of specimen X, which is the sisal.
A doormat.
Question E. To which group of plants does the plant of specimen Y belong?
Flowering plants. This is because it has parallel venation.
Question number 13. You are provided with the following: a part of stem marked X, a laser blade or a scalpel, a handle, a 30 centimeter ruler. Using a laser blade or scalpel, cut a cross-section through the middle of specimen X and observe with a handle. Question A. Draw the cross-section of specimen X and label any 2 parts. Specimen X is a cassava stem or a sugarcane stem.
On observation, we have observed the following. Number 1, phloem.
This is the part of a stem that stores food.
Number 2, xylem.
This is the part of the stem that stores water.
The following parts were observed by using a hand lens.
Question B. Measure the longest axis of cross-section of specimen X and record your results in millimeters.
By using a ruler, measure the longest axis of the stem.
And calculate the magnification of your drawing. Show your working.
By using magnification formula, magnification is equal to length of the drawing divided by length of the specimen.
Question number 14. You are provided with the following: specimen P, which is a lemon; specimen Q, which is a tomato; and a 30 centimeter ruler. Question A. Draw specimen P, which is a tomato, and label any 3 parts.
Number 1, epicarp.
This is the outer part of the tomato.
Number 2, seeds.
This is the part which is stored for replantation.
Number 3. Placenta.
This is the part that is found at the middle of the tomato. Question B. Which of the following specimens can withstand dry condition? And give the reason for your answer.
Specimen P, which is a lemon, because it has cells that absorb more water.
Question C. Measure the length of specimen P, which is the tomato, and give your answer in millimeters.
By using a ruler, measure the longest axis.
Question D. Calculate the magnification of your drawing.
By using magnification formula, magnification is equal to length of the drawing divided by length of the specimen.
Question number 15. You are provided with the following food samples. Sample X, which is gaiwa or maize flour. Sample Y, which is groundnut flour or singilo. Iodine solution, 10% of sodium hydroxide solution, 1% of copper sulfate solution, a white paper, and 2 test tubes. Question A. On sample X, which is gaiwa or maize flour, and on sample Y, which is groundnut flour or singilo, perform a protein test and record your findings in the table.
Take a spatula full of groundnut flour and put the flour in test tube A. Secondly, take a spatula full of maize flour and put the flour in test tube labeled B. Add water in both test tubes and shake. Add sodium hydroxide and copper sulfate solution in both test tubes, then shake. Observe any color change. On observation, the color of copper sulfate changes to violet in test tube B, while in test tube A there will be no any color change. The color of copper sulfate remains blue.
Question B. Draw a conclusion from this experiment.
Purple or violet color indicate that there is presence of proteins in test tube B, which contains groundnut flour, while in test tube A, which contains maize flour, there was no color change. This indicates the absence of proteins.
Question C. Which food sample contains food nutrients that will be suitable for a baby growth?
Food sample Y, which is groundnut flour, because it contains more proteins that are suitable for body building.
Which of them is suitable for supplement in a child with marasmus?
Food sample Y, which is groundnut flour.
Question D. Explain difference in energy content between food sample X and Y.
Food sample Y, which is groundnut flour, has much high energy content compared to food sample X, which is maize flour.
You are with me, Madam Rita and Sir Godfrey Malata, and here is our contact 09929725544. I will repeat 09929725544. Share this from Chiriba to Sanje. Thank you everyone, and wishing you all the best and good night. Thank you.
Zikomo kwambiri Madam Rita, zikomo kwambiri omvera nonse from Chitipa to Nsanje. Mnali ndine Sabu.